Hi, everyone. You're listening to episode 654 of the People Behind the Science podcast. This week, we are rebroadcasting our interview with Dr. Malia Gehan, which was originally released on June 20th of 2016. Listeners, Malia is working to improve crop plants by incorporating traits from hardier weedy plants to try to make the plants we eat more resilient in response to temperature stress or other environmental stresses. And Malia shares more about her work and her life in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am thrilled to introduce you all today to Dr. Malia Gehan. So, Malia, thank you so much for joining us today. How are you?
Thank you for having me. I'm great.
Wonderful to hear. Well, we are excited to hear about your journey through life in science, but I want to start by introducing our listeners to you with a little bit of background. So, listeners, Malia is a new assistant member at the Danforth Plant Science Center. She received her undergraduate training in biology from Willamette University and her PhD in plant biology from Michigan State University. Afterwards, Malia was awarded a National Science Foundation Plant Genome Initiative Postdoctoral Fellowship working at the Danforth Plant Science Center, and she subsequently worked as a research scientist there before accepting her new position. And now Malia is here with us today to talk about her experiences in both life and science. So, Malia, we are really looking forward to getting to know you, both as a scientist and also as a person. So I want to start by learning a little bit about you and your life outside of science. So can you tell us what you do in your free time?
Sure. So in my free time, I try and spend as much time as I can with my husband, who's also a scientist, and with our two cats, who are adorable. And we spend a lot of time sort of lounging at home and walking around in our neighborhood. We live in a really nice neighborhood just south of Botanical Gardens and a beautiful park. We like cooking and going to the movies and watching TV and sort of de-stressing from the stresses that sometimes occur when you're a scientist and can't turn off the life, because science really is.
It's always in your mind. Yeah.
It's not a career, it's a way of life. And he understands that because he's a scientist as well. And so I'm really lucky.
Well, that is awesome. It sounds like you guys are able to escape the stress of lab a little bit with all these different hobbies and things. So wonderful to get a window into your life outside of science. Now, I'm curious, say you're at a dinner party. How do you describe what you do at a very high level to someone who has no idea about what your field is about?
The big question that I'm trying to answer is how to improve crops to temperature stress. That's the big question that I am interested in solving. And to do that, the approach I'm taking is to look at natural variation. Nature and evolution and all the different environmental constraints that we already have out there have created plants that are really resilient in a lot of different environmental conditions, but they're not necessarily good to eat. So how can we take those characteristics from weedy plants and put them into domesticated crops?
Interesting. Well, I think these weedy plants have a lot of traits that would be great for regular domestic crops. I think they're one of the frustrating things about them is they're so durable, right?
Right. You'll see dandelions growing out in the cracks of sidewalks, and there's not very much water there. So how can we get those traits into crops that are less drought resistant, like maize?
Very cool. Well, wonderful to get a little bit of insight into your work, and I'm excited to chat more about it as we go through our interview here. But I want to take a pause here and talk about motivation. I think, Malia, as you're talking about your work, you can sense the passion in your descriptions of it. So I think it sounds like you are bursting with motivation. What inspires you or what motivates you? And maybe do you have a particular quote attached to this motivation?
I have been thinking about this motivational quote question, and I couldn't come up with an actual motivational quote, but something we say often in the lab is garbage in, garbage out, which is just a very impolite way of saying measure twice, cut once.
Sure.
So that motivates me because I'm often a careful person, and I sometimes feel the pressure of going faster. But then I remind myself, if you're careful in the beginning, you're more likely to be satisfied with your results in the end because you took all the precautions and steps that you needed to get the results that you think are the best, that you feel the safest with.
Definitely. And I think all those efforts on the other side are wasted if you're not really careful in those initial inputs of your data.
Yeah. And then the opposite quote to that, which is a piece of advice that a mentor gave to me, which is don't let the perfect get in the way of good, which is sort of the opposite. Mm-hmm. But it's something that I often have to tell myself because I have to remind myself, are you being a perfectionist? Is this really good enough?
Definitely. Well, I think it's a delicate balance between the two. So how do you manage it? How do you decide what is good enough versus what is you're not being too careful?
I rely a lot on my gut and my peers. I'm so lucky that I'm surrounded by really great scientists here at the Danforth Center who are inclusive and who are willing to give their expertise and opinion on each other's research. There's a lot of peer-to-peer mentoring here at the Danforth Center. It's a really special place to be.
Fantastic. Well, now that you bring up these peer mentors, I think the people who are able to mentor you are really important throughout your scientific career. So are there particular people that you can point to, maybe from an early on standpoint, who really served as role models or mentors to you then?
So I went to Willamette University, which is a great liberal arts school in Salem, Oregon. And I had my first real experience doing research in the lab was with Gary Tallman, who's about to retire. And he's an amazing mentor because he basically trusted a sophomore in college to do research in the lab pretty independently. And he also takes the time out of his busy schedule to teach people protocols late in the day. And it's a passion for him. And I think that's transferred to the people he mentors. And so I was very lucky to have him as a mentor early on.
Certainly. Well, wonderful to hear about one of these people who really inspired you in your college years. So can you tell us a little bit about how you got interested in science in the first place?
Sure. I was really a late bloomer to science, I think. But as I've reflected on how I've become a scientist, I now realize that sort of just like plants, I'm a product of both my parents and my environment. I was born in San Francisco, but moved when I was really young to Hawaii, where my mom is from, and I was raised there. Both my parents are artists, and a big part of why I think I'm a scientist today. First reason is actually because most kids like to rebel against the occupation of their parents, which is probably why I was later attracted to science. But art and science are both really creative pursuits. And my parents gave me a strong appreciation for both creativity and observing the natural world. So one thing they had me do when I was young was that they would have me draw things upside down, in part because they were teaching at that time. And so they were practicing lesson plans on me. But often you can get a more unbiased perspective on an object because you want to draw, if you draw it in that way. And science is often like that. You want to get a different perspective on a question. But when I was in high school and when I was transitioning to college, I really didn't know what I wanted to do. I liked history. So as a freshman, I decided to be a history major, but it was sort of a default. So I graduated from Willamette University, like you mentioned earlier, but I didn't start there. So my freshman year, I actually went from Hawaii to an all-women's college, Mount Holyoke, in South Hadley, Massachusetts. It was a major culture shock. It's a great school, but I think I knew right away that it wasn't going to be a good fit for me. So I decided to transfer pretty quickly, but after my first year. And because I knew I was going to transfer, I knew that I wanted to get some sort of standard class requirements out of the way. And so I decided to take a science for non-majors course. I had never considered even going into biology as a major, but in many ways that science for non-major course was this sort of low pressure way of being introduced to biology in a way that I could just really enjoy it. So there was a research component to the class. We had a couple week long experiment where we were applying GA to pea plants and having to make observations in the lab. And the lab was open, so we could do the lab on our own time. And this was a big revelation for me because I realized I could do things at my own pace. And I remembered in high school never liking lab because I always felt like I was rushed to finish the work within this hour-long period that we had.
Right, me too.
And I hated that. I did not like feeling like I had to rush to finish something because I enjoyed it. And from that point, I knew that biology was something that I wanted more of. So when I transferred to Willamette that following year, I became a biology major. So in many ways, I jumped in. I took one class and then that changed my perspective and completely changed my major. It was a drastic change, but I was really following my gut and I knew that it was right. So because I went to Willamette, which is a small liberal arts school, there were actually quite a few undergraduate research opportunities. So I approached Gary Tallman about research in his lab. I liked the idea of working with plants because I didn't want to have to kill animals. And I volunteered for a semester and then got what was equivalent there to a summer RAU position. And I loved being in the lab. I think most scientists are scientists out of this love because it's a demanding lifestyle. Plants don't sleep.
Right.
Sometimes you can't sleep either. And so at the time, the Tallman lab studied heat stress and guard cell protoplasts. And the idea of temperature stress was really interesting to me, in part because of where I grew up. So in Hawaii, you can really hardly say that there are any seasons. There's really very little difference between winter and summer.
It's just beautiful year round, right?
Just beautiful, yeah. So from going from Hawaii and then going to Massachusetts and then going to Oregon, I just realized that plants are surviving outside in conditions that I definitely could not. And so that's really why that research question intrigued me. And doing research in the Tallman lab was great. He's an amazing mentor. And I knew that I wanted to do more research, so graduate school was just a really logical choice. And I did rotations in other areas of plant science. I did a rotation in a plant pathology lab, in a signaling lab at Michigan State. But ultimately, I went to Mike Thomas's lab to study interactions of cold stress in the circadian clock in Arabidopsis, because I was still very interested in temperature. And Mike was an amazing mentor and really led his people follow the projects as they see fit while still kind of keeping us on track, which I think all graduate students need.
Definitely.
And after I finished my thesis with Mike, the area of temperature stress still really intrigued me, but I realized that I needed to sort of branch out. And so I applied and was lucky enough to get an NSF postdoctoral fellowship at the Danforth Center to study temperature stress. But this time, instead of in Arabidopsis, I'd be moving to grasses. I could have kind of kept in that lane of molecular biology and temperature stress, but there was an opportunity to start working in high throughput phenotyping because there was a platform being built at the center. It was really special. And so I volunteered. And the field itself of high throughput phenotyping is very, very new and in many ways still very much like the Wild West. But I saw it as a way, again, of jumping in to something new and as a way to bring a fresh perspective to the question of temperature stress that I'm still very, very interested in and will continue to be interested in for, I'm sure, many, many years.
Definitely. Well, wonderful to hear more about your story, Malia. And I love so many of the points you brought up. I know I'm a student who came from a small liberal arts undergraduate school, and it is a phenomenal experience. There are a lot of great opportunities at those kinds of schools.
I feel like there's a lot less pressure. We both, I guess, weren't in an environment where it really was the undergrads doing the research in these labs.
Right.
So you're kind of the ones doing the stuff.
Right, exactly. And you're working side by side a lot of times with the faculty, whereas you don't see that necessarily in a lot of larger research institutions.
Yeah. So Gary trained us to do cell culture himself, and that would probably not happen in an R1 institution. So it was really special to be at a small liberal arts school.
Well, now you hinted that there's still a lot of work to be done in this high throughput phenotyping. And I know that's something that you're kind of working on right now. So can you tell us about one of these projects that you have going on that you are just so excited about?
I am very interested in phenotyping quinoa for a very specific reason. And that's because it's been reported to be very, very tolerant to abiotic stress.
Interesting.
So it can grow on salt plains and also at very high altitude where there's very low temperature. And it also grows in subtropical regions. So it's a plant with a large range of environments that it grows in. And so I'm interested in phenotyping those traits in a lot of lines that have been collected throughout South America. That's where quinoa originates from.
Gotcha. Very interesting. So it sounds like you are particularly interested in temperature as an abiotic stress. Do you look at other abiotic stresses?
I do. So cold can be a type of drought stress because if you freeze water, it makes water less available to the plant. And so there's a lot of overlap in those gene networks and pathways. And so when we look at cold, we often look at drought. And because I've also done work in the past on heat stress, I'm interested in the interaction between these stresses.
Well, this is really cool, Malia. So I'm curious, do you get to go out in the field at all and collect samples, or do you work primarily in the laboratory?
I work primarily in the laboratory, but I am really, really hoping that I will get to do collections.
Fantastic. Well, exciting to hear more about the wonderful facilities it sounds like there, as well as some of the research that you're doing. But I think we've made your career in science sound like a little bit too much fun, Malia. So I'm going to make you talk about some of the challenges, the struggles, the stresses, because I know these are real and these are a problem in science. And these are things that you have to deal with and overcome as you go through all these different steps of your career. So Malia, can you tell us about one of your own challenges or failures and just how you worked your way through it to be successful?
So working on this phenotyping system, we came to the conclusion that we needed to create new software to analyze the data. And I'm not traditionally trained as a computational scientist, but I became one. Mm-hmm. And that was through a lot of stick-to-itiveness and gumption. and I had to gain the knowledge on how to do these things. But it was hard, and we accomplished a lot. And I worked a lot with Noah Falgren, a fellow researcher here at the center, and we helped push each other along because we were both new to this area of image processing and phenotyping. So that was a struggle, but because I had this group of researchers here to support me, we really got through it. The second half of that story is that it took a long time for us to get the work published, which is, I think, a common problem for a lot of scientists. It can be very difficult to prove to other scientists that your work is important. And it took a long time to get that work published, but we did. And one success or validation that came out of that whole project is that we're now at the point of developing that software where other people are saying they're using it and that they like it, and that we actually have received grant money to even extend it further because people like using it.
Well, that is awesome. Well, congratulations on turning one of these challenges into a success. And I think being on the cutting edge of science, you kind of face similar problems like this a lot. You're always learning, you're always trying to pick up new skills, but there are often not a lot of good resources out there yet for you to learn these things from. So
exactly.
What helped you guys move along and pick these skills up?
I've come to the realization that programming is a lot of advanced Googling.
I attribute all of my programming knowledge to Google as well. So
exactly. And coming to that realization and accepting that has really helped, and being comfortable enough asking for help. I think a lot of people are scared in science. I mean, I struggle. I think a lot of people struggle for imposter syndrome, and you don't want to appear that you don't know something. But often I think you need to know enough to know what you don't know. And because there's so many great scientists here at the center, we have this sort of cohort of people that we can ask who have expertise and experience in all different areas.
Certainly. Well, wonderful to hear about how you work through one of these challenges. And I think being resourceful is part of the life of a scientist. Like you said, sometimes you just have to suck it up and accept what Google or Wikipedia or whatever might be telling you.
Exactly. I mean, we often have to work within the constraints of funding. So we have to get creative. And that's one of the big things that I've gotten more and more aware of as I've gone along. So that's a big reason why we started a maker group here at the Danforth Center. It really started because we had that large robotic phenotyping facility, which booked up really fast. It's actually booked up through 2019.
Oh my gosh, wow. Funded
projects through 2019, and they have experiments going all the way out to 2020. And we realized we can't be waiting for an experiment on the system, even though those experiments are amazing. We need other ways of phenotyping plants. And so we started to make these low-cost systems to complement the data that we were getting off the phenotyping facility. So maybe we can pre-screen plants, and then that way we're putting the most interesting plants we know about on the phenotyping facility. So we use low-cost cameras and computers. So there's a company that makes low-cost computers. They're called Raspberry Pis.
That's amazing. I love the name.
Yes. It sounds delicious. They're not very good to eat, but they have ones that cost $5 and they have the ones that cost $35.
Oh, wow.
They're fully functional computers, and you can hook these camera modules to them. So the camera module costs about $30. And so we have something that's very cheap, and now you can record image data, say in a growth chamber or in a greenhouse now over time, and get similar sorts of data to the ones that we're getting from the Bellwether facility, but we're getting that in sort of different locations.
Well, I love this creative solution to resource constraints. And I think you're seeing this more and more in science as people are coming up with lots of great ideas, but there's always that restriction on resources.
Exactly. We have to do more with less.
Certainly.
Just like we need more plants with less resources, we need to do more science with less resources.
Absolutely. Well, thank you so much for telling us more about this Makers facility, as well as some of these challenges that you faced in science. But we've touched on so far a couple of your successes, and I love celebrating the successes in science in every form, the big wins, the little victories along the way. So Malia, can you tell us about another one of your own successes that really meant a lot to you?
One recent one, which has a little less to do with science and a little more to do with outreach in science, is a grant that we got from the Raspberry Pi Foundation. So again, that company that makes those low-cost computers, we got a grant to have some fun educational events feature Raspberry Pis.
Awesome. So are these with local communities? Is this with the youth? Are there particular populations that you're targeting for these outreach events?
Yes, and yes, basically.
Awesome.
So we put something called a raspberry jam or raspberry Pi jam on,
Amazing.
which is basically fun family event where people from the center show off their projects that they use Raspberry Pis with, but it doesn't necessarily have to be a Raspberry Pi project. It can be any fun project that uses technology. And we bring local robotics groups in. So St. Louis has a very strong robotics community. The first robotics finals was just recently here in St. Louis.
It was, yeah.
I got to be a judge at the regionals, which was really fun.
Really?
Yeah.
Oh my gosh, that's awesome.
That's another great thing about the St. Louis community is that there's so many people that are really connected. So that actually was a connection I made because we had this maker group at the Danforth Center.
Very cool. So tell us a little bit more about these raspberry jams. So what is your role in it? How do you interact with people?
We show people how we use these low-cost computers in our research. And I think they're normally used for hobbyists. So a lot of kids are actually familiar with them because they like to play Minecraft on them.
Okay, gotcha.
But now we're showing that we're using them for legitimate science. And I think the kids get really excited because they see that this is a tool that they normally use for fun that can be used for research. It's a way of sort of sneakily showing them how science can be really, really fun.
Definitely. I think it's cool that you're using such an accessible piece of equipment, right? Because you can show people things on an MRI scanner, but it's not something they're ever going to be able to use in their own home, in their own lives.
Exactly. So there's some great programs. We have a great educational director, Terry Woodford Thomas, who has several tech trunk programs. And another part of this Raspberry Pi Foundation grant was to create tech trunks that incorporate these Raspberry Pis so that they can be taken to schools.
Well, that is really, really cool, Malia. So thank you so much for sharing your interest in these outreach events, as well as congratulations on getting this grant to allow you to be able to do them.
It's a really great opportunity and really fun for scientists here at the center as well.
Definitely. So in addition to some of the work that you do in the laboratory, some of these research questions, one thing that I do also love to chat with people about are books. I know I am an avid reader myself. I'm always adding things to my to-read list faster than I can check them off, of course. But Malia, is there a book that you've enjoyed and you want to recommend to me and our listeners to add to our lists?
I actually just finished reading Every Other Thursday by Ellen Daniel. And it's a book about stories and strategies for women scientists, but really it can be applied to really any scientist throughout their career. They had this peer mentoring group that's been going on for over 25 years. And it's really the stories that have come out of support and trials and tribulations from that group of women scientists.
Well, this sounds like an awesome book. Believe it or not, I have actually never heard of it. So
Really?
I need to look into this.
Yeah. There's one particular story in there is about women not getting tenure and how they worked through that.
Mm-hmm.
It was really refreshing to see not just the ups, but also sometimes the downs. And it's a bit motivational to hear that because these are amazing scientists going through the harder times.
Right.
And that's really inspiring for someone who's pretty young in science like myself.
Definitely. Well, I am going to add this to my reading list. I'll put that out there on our website as well for our listeners to find. And Malia, we've talked about a lot of different aspects of your career, but I think we haven't yet touched on travel. And I think travel is a big part of the life of a scientist for conferences, training, meetings, whatever it might be. So can you think back now on all the places you've been for science and tell us about your favorite?
I have a hands down answer for this.
Excellent.
I got invited to go to France
Ooh.
for a conference called PhenoDays, which is a phenotyping conference. And number one, it's a great conference because it's really focused on the technology and the tools. You can really see how it can impact your own research. And so in that way, it's very interesting. And then it was in this beautiful location. The last night of the meeting was in a French chateau.
Oh, wow.
And there was delicious food. So anywhere there's delicious food, you basically have me. And it was in France. So this is the hands down winner.
So where in France was this?
It was in the Burgundy region.
Gotcha. I've only been to Paris, but it is beautiful. Everything I've seen, the pictures from people who've been to other places, looks gorgeous.
So beautiful. It was at a walled city.
Oh, wow.
And so there were wineries and we got to go on a mustard tour on one of the days that were free. Mm-hmm. So it was really fun. And I was with colleagues. So it was extra fun because I knew a couple people that were also at the meeting.
That's awesome. So do you speak French at all?
Oh, zero. I just basically pointed at the croissants.
Right. My favorite thing is when I don't speak the language, just point to a random thing on the menu and see what it actually turns out to be.
We use a lot of World Translator, which is this app that you could put your phone over and actually see the translation pop up.
Oh, gotcha. I do the menu roulette approach, but it hasn't failed me yet.
Yeah, sometimes the translations, though, were still like menu roulette.
Right.
Because it was like cousin of duck or something was one of the items on the menu.
Well, this sounds like an amazing trip. And I think this is one of the things that I really love to share with people out there who might be considering careers in sciences. You do get to travel, you do get to see the world and meet colleagues internationally who are all excited about the similar questions in the same field.
Yeah, and phenotyping in particular is a really international field. A lot of colleagues in Belgium, in Australia, Germany. And so Skype's amazing because we can connect with them on a regular basis. Mm-hmm. And that means that we get to go to a lot of international meetings, which is always fun.
Definitely. Well, great to hear more about your travels and kind of in talking about your colleagues, your mentors. I think we've really been hinting at the human side of science throughout our discussion today. And I think there are so many wonderful people and personalities in science. I think it's hard not to develop these quirky traditions and just kind of fun and fond memories that you share with people. So Malia, can you share with us today one of your favorite fun or funny memories in science or a quirky tradition you experienced?
I don't know if it's quite a tradition yet because my lab is very, very young. But myself and a colleague, Katie Greenham, who is currently a postdoc in Rob McClung's lab at Dartmouth, she'll come to visit the Danforth Center. And we'll often have to do a very intense period of lab work whenever she's here because we're trying to get things done. And so whenever she comes, we have this tradition of buying just a ridiculous amount of delicious food and eating that food whenever we have a 5-minute incubation.
Amazing.
So it's like scheduled snacking. So that's the tradition that we have sort of started and I hope will continue throughout the coming science years.
Well, this sounds like a wonderful opportunity for a reasonably spaced snack marathon throughout the day then.
We keep our energy up with all the snacks.
Of course. So Malia, what is your favorite snack of choice then?
Ooh, we like these inner peas, like freeze-dried peas.
I've seen them before, yeah.
From Trader Joe's, yeah, and then we dip them in hummus.
Ooh.
Those are very good.
Solid choice, solid choice.
Semi-healthy.
Of course. It doesn't count if it's a vegetable. Right?
Exactly.
So that was funny thinking back to graduate school. It was one of the reasons I chose to work in a lab that worked with human participants. It's not a wet lab at all, no blood, no nothing, because I could snack throughout the day at my desk. That helped me make my decision of which lab to choose.
Yeah, it's good because we'll have to move into an office because we can't obviously eat in the lab.
Of course.
And that's good because we get up and then we sort of have this timed break.
Right. There's an end to the snacking.
Yes. Controlled snacking.
I like it. Well, that is a fantastic tradition to start, Malia. So it's been great to hear about some of your experiences in science, as well as some of the people that you've been able to work with and some of the questions you're able to answer. But I want to know today, if I gave you everything, all the funding, staff, technology you would want, feasibility is not a problem. Malia, is there one question that you are just dying to know the answer to that you would want to tackle first?
I've been thinking about this mainly because my lab's getting started and I'm trying to figure out which are the big questions I want to tackle first.
Sure.
And one of the outstanding questions in the field of phenotyping obviously relates to global food security and security in general, because food security is really general security. And that has to do with how we can more effectively translate our knowledge from the lab to the field, because we can gain a lot of knowledge in the lab and we can also gain a lot of knowledge in the field. And we need to be able to bridge that gap more effectively. So data across scales, how can we more accurately or efficiently translate data across scales?
Definitely. And I think this is a challenge that we're seeing not only in the plant world, but also in lots of other fields of research. I know I work in a more clinical research field, so it's really hard for us to translate the same thing from the laboratory to the clinic. So do you have any ideas or ways that you would go about trying to do this?
Right. So we're trying to attack this problem head on by measuring all the things. We really need more information, and that information needs to be collected in a way that is as close to each other as possible. So often I think there's a disconnect between what we see in the lab and what we see in the field because those measurements are collected very, very differently.
Sure.
We need tools that are able to bridge that gap. So tools that we would use in the lab and then take it out to the field and vice versa. And I think we're starting to see that, but that needs a lot of development still. That's the question I would go after if I had unlimited resources.
Well, wonderful to hear about one of these big questions that you are working your way towards answering. And I think it is a really important question. And I think these big questions are the ones people get most excited about, right? You build your lab around the big question and kind of decide what little pieces you can contribute towards answering it.
Right. And even though people are working on this question, this isn't a question that's going to be solved overnight. It's going to be solved with lots and lots of people, lots and lots of groups, these huge interdisciplinary projects. It's not going to take just biologists. It's going to take engineers. It's going to take physicists it's going to take a lot of people.
Definitely. Well, thank you for dreaming big with us today, Malia, with your research questions. And I know throughout our discussion today, we've talked about some of the mentors, some of the people along your journey who've helped you along the way or made a big impact on you. And I think these individuals are wonderful for providing advice during maybe difficult times or just to keep you motivated and inspired along your journey. So is there one piece of advice, Malia, that you can pass forward to our listeners that you think is just the best piece of advice you've ever received?
I think I've mentioned this actually earlier in the interview, and it's because of my own personality tics where I'm a type A perfectionist. And it's that whole thing about not letting the perfect getting in the way of the good. And to remind myself that people need to see what we're doing in order to find value in it. And so they need to see your progress along the way. So you need to let people see your progress and not be restricted by your criticisms of yourself.
Certainly. I think not being afraid to show people a work in progress and say, this is where I'm at now, but it's not done.
Exactly. I think we're scientists trying to figure out what's the quote unquote truth, right? So we're naturally skeptical. And so we're skeptical a lot of ourselves. And we often are too critical on ourselves and need to sort of get the information out there.
Absolutely. Well, wonderful advice for our listeners, Malia. Is there any other last piece of advice or maybe a last note of inspiration that you want to leave all of our listeners with today?
I would just encourage everyone to go for it. If you want something, find opportunities that are unusual. The Raspberry Pi Foundation grant was something I found off a tweet.
Really?
Yeah. So they tweeted that call and I saw it and I said, well, why not? Why not apply for that? I'm often finding that there are very interesting opportunities out there. You just have to look for them. It can be hard, but you have to just go ahead and do it.
Definitely. And I think keeping an open mind is a big part of that. Not just seeing that and kind of dismissing it, but thinking, why not me? Why not apply for this?
Right. Exactly.
Fantastic. Well, wonderful message to leave our listeners with today. Malia, can you tell them where they can go if they want to learn more about you and the amazing work that you do?
You can always reach me at Twitter. My handle is Malia Dong, MALIADONG. That's my maiden name. You can also reach me through email. My contact information is on the Danforth Center website, danforthcenter.org. I also have a lab website, Gehan-lab.org, and I'm also hiring. So if you are interested in finding out more about my research or working with me, please don't hesitate to contact me. I'd be very interested in hearing from any scientists interested in temperature stress and phenotyping and all of the fun things.
Fantastic. Well, I know there are probably a lot of scientists out there looking for jobs. What type of positions are you looking for?
I am hiring at all levels, but I'm interested in hiring a postdoc right away.
Awesome. Well, listeners, definitely check out the websites Malia mentioned, follow her on Twitter, and contact her if you are a postdoc or other scientist interested in the field looking for a new position. So Malia, thank you so much for joining us today.
Thank you.
It was a pleasure to chat with you. Listeners, a pleasure to have you here as well. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.